All right, hello everyone. This is Dr. Rossler. We're going to be working on a remote lab for week 7. And week 7 is actually a pretty interesting lab. It's DNA biotechnology. And we're going to be kind of exploring some of the concepts that are used in modern DNA fingerprinting. And DNA fingerprinting is a way that we can determine uh, a specific identity of an individual based upon their DNA. So just like you have different fingerprints that make you unique or are uniquely associated with you, your DNA also has a unique fingerprint that is uniquely you. All right, so we're going to be looking at that in relationship to one specific gene today. And yeah, this is often used in forensics as well as in disease identification. So if we're trying to determine what the probability is that a patient carries the gene, for instance, for like BRCA1, which is highly associated with breast cancer, essentially some DNA could be taken from the patient and we can amplify that gene and then determine whether or not they have the quote-unquote good gene or the quote-unquote bad gene version by doing a DNA fingerprint of that region. All right, so that's what we're going to be doing today. So we're going to be doing it specifically for a gene in TNF-alpha, tumor necrosis factor, which is involved in your immune response. So it's kind of cool, right? So some of you might have one version of the TNF-alpha gene and some of you might have the different version. And so we're going to do this little experiment to see how it is virtually. All right, so let's get into it. So first things first, when you're doing any sort of DNA work, you need to get the DNA, right? Makes a lot of sense. So DNA, you guys know, is nucleic acid. And let me draw some stuff on here. DNA, of course, is made up of A's, T's, C's, and G's. And each of those nucleotides, A's, T's, C's, and G's, is interconnected with phosphates. Now, phosphates are PO with a -3 charge, and there's 4 oxygens, right? And so what we're meaning by this is that those oxygens, let me just draw this out a little bit better, each of those oxygens has a negative charge. All right, those 3 oxygens. And so those are all along the length of the DNA helix. So there's a lot of negative charges throughout your DNA. And so what that does is it makes DNA a very negatively charged molecule. And I'm going to write that down because it's really important. DNA is inherently negatively charged. And we use that a lot. And it's negatively charged, again, because of those phosphates in the backbone. All right. The DNA is also encapsulated, as you guys know, in a couple places throughout the cell. If we're talking about an animal cell such as ourselves. And the main place that we have our DNA, right, is in the nucleus of the cell. But we also have some mitochondrial DNA, right, that we inherit typically from our mothers. And if you're a plant, you have some chloroplast DNA. All right, so there's in most eukaryotic cells, again, all eukaryotic cells, we're going to have nuclear DNA, mitochondrial DNA, and then the plants, right, are going to have the chloroplast DNA. All right, now we've got 3 little sacs or 3 little membranes that are encapsulating or hiding the DNA, and we need to get to them. We want to get to all of this DNA depending on what organism we're extracting DNA from. And one of the first things we need to do is we're going to take a sample of the cells. You don't need very many cells, right? You can actually have 1 cell and get the DNA from, or of course, if you have more cells, you're going to get more DNA. But you would take a sample of those cells. When we do it in humans, we often do it from their cheek cells. So you just take a toothpick and they kind of rub it along the inside of their mouth. Again, you could take a hair sample. You could take, other samples that you could get from an individual, and you would take that sample and you would treat it with a little bit of soap. And let's think about what the soap's going to do, right? So the soap is an amphipathic molecule. And the reason why soap gets the grease off of your pans so well is because soap is really good at emulsifying fat. And what that means is it kind of breaks it down. So what the soap is going to do is it's going to break down the cell membrane into tiny little bits, as well as the mitochondrial membrane, the nuclear membrane, and even the chloroplast membrane. So all these little areas where we have DNA are going to be able to be opened up, essentially. All right, so that's what the soap does. It emulsifies those phospholipid membranes that are, again, around these different structures. The second thing that you're going to add if you do this is a lot of salt. All right, so we typically are going to use sodium. Right, so you use sodium, and what the sodium does is sodium is a positively charged cation, and it's going to be really, really attracted to the negative charges in the phosphodiester backbone. All right, so you're going to have all these little positively charged ions now that are going to form an ionic salt bridge, right, with those negative charges in the phosphates. And what that does is now DNA is no longer negatively charged. It's kind of neutral in charge. All right. So again, what the salt does is it gets rid of that inherent negative charge. Now, why do we care about that? We care about that because the ultimate goal of DNA extraction is to get the DNA out of the water, right? And by getting rid of those negative charges up here, we're actually making the DNA less soluble. It's still soluble, but we're making it less soluble. Hopefully that makes sense. The third thing we're going to do is we're going to add some protease. Now what proteases are, as you can imagine, is they're enzymes, right? They're enzymes that break down proteins, right? Just like the name implies. Now why do we need to break down proteins? There's a couple reasons, right? You have proteins that are in the cytoplasm of your cells that degrade DNA. So you might have some proteins in here that are called DNA, sorry, DNAases. They're literally called that. And DNAases' job in life is to degrade DNA. So the proteases break down the DNAases, which is really nice. So then our DNA is going to be protected. Now also, why is this important, is that you have these other proteins that are bound to your DNA called histones. And histones are really good at winding up the DNA. So it's really, really tight and congealed in this little area, the little sacs, or the nuclei, or the mitochondria, the chloroplast, right? And again, what the proteases are going to do is they're going to essentially get rid of all that. They're going to get rid of these histones, degrade them, right? So now that the DNA kind of whoosh, comes pouring out of those it's just much more accessible, right? Hopefully that makes sense. So they're going to break down those proteases, like DNAases, and they're going to break down the histone proteins that normally wind up our DNA. So now it's all unwound, right? It's going to be much less condensed than it previously was. Now, it's really not that hard, so you let those things happen. And then the last step in DNA extraction is typically to add cold ethanol. And so if you do this at home, I put here actually the protocol of how you could do this at home if you do want to repeat this experiment. If you have a couple strawberries sitting around at home, strawberries are great to do this with. You use rubbing alcohol and you just put it in the freezer for a while. And why do we use alcohol specifically? Ethanol, it's because DNA is not soluble in ethanol. And so by adding it, we're going to get the DNA to kind of come out of solution very, very quickly. Excuse me. So here's the protocol for doing this. If you want to do the same experiment at home, it's really fun. If you have kids especially, I encourage you to do this. Again, you would just take a strawberry or two, just put it into a little sack. I've actually done this for non-majors who we don't have a lab for, and they get a real kick out of it. So you would take a strawberry and just kind of mash it up in a Ziploc bag. You add a little bit of soap, just literally like a pinch of soap and a little bit of salt, water into the solution, and you kind of swish it up and make kind of like a nasty little congealing mix. Then you would pour it through essentially a coffee filter, right? So you're going to have all the little big bits of strawberry that don't get mashed up on top, and then you're going to get this sort of strawberry juice with soapy water and salt water down here. And then what you're going to do very quickly is you're going to add some ethanol, some just really, really cold ethanol. And what you're going to see really quickly happening is that the DNA precipitates out, just like what we talked about. And so this is what you do if you work in a lab to a very smaller extent, right? You have like these little tubes that we do it in if we have just a small amount of DNA to start off with. But it's really, really fun. All right, so there you go. Hopefully you try it. All right, so what we're gonna do now is do this virtually. And so what you can do now, if hopefully I can get out of this, let's see, I'm gonna end the slideshow. All right, we're gonna get rid of that. All right, so what I'm gonna have you guys do is go into your textbook. All right. And if you go into your textbook online, your McGraw-Hill Connect, all right, why don't you can pause this until you get that get that opened. You should see week 7 remote lab part 1 and part 2. All right, so hopefully you guys are all there. And what you're going to do is go ahead and open up part 1, as you can imagine, and you can read a little bit about, this is very much like what we just talked about. The key concepts. Again, you guys can always go back and see this sort of introductory material as you're doing the lab. All right, and it's going to discuss how this experiment works. So what you're going to do is what we just described, right? You're going to be using strawberry extract. And you're going to get some of that strawberry extract out. You're going to mix it with some salt water, and you're going to hopefully then, here's the proteases. The protease that you're going to be using is meat tenderizer. Meat tenderizer is, you don't need it for this experiment, but it helps to make it work better. But again, it is just a protease, and it's going to help to degrade those proteins, those histones and DNases that we talked about. And here is the cold ethanol that we talked about that you're going to use at the end. So I'm going to have you guys go forth with that and get the whole experiment done and save your data at the end. All right, you can pause it here and do the experiment. And again, if you ever get stuck, you can hit Control Alt K, I think it is, or Control or Command Alt K to kind of skip through. But I think you guys can get through it all. All right, so hopefully you guys got all the way through week 7 remote part 1. I realized I may not have shared my screen at that last bit, so hopefully you guys all figured that out. All right, so what we're going to do next is, before we do part 2, let's talk about the theory of part 2. And to do that, we're going to go back to our PowerPoint presentation. All right, and hopefully I'm sharing this. I always have to check. All right, good. And what we're going to see is, so part 2. So now we've extracted the DNA. So let's imagine we did this out of you. We got a couple cheek cells. And now what we want to do is we want to do some experiments on that DNA to determine what your genotype is. All right. Now, firstly, one of the things you do in this scenario is you do PCR. So PCR is pretty commonly used in DNA fingerprinting, or any of these experiments that, you probably heard a lot about PCR. It's been talked a lot about. There's certain COVID tests that use a PCR amplification to determine whether or not you have the virus. And so PCR stands for polymerase chain reaction. And the reason why it's been really handy in COVID analysis is that we know what the gene is, that the genes that are associated with the virus. And what you do, you might just have a small amount of the virus in your body. And what they do is they take the DNA sample from your body, specifically right in your nose, your sinuses, and they try to amplify the viral genes to see if you have them. All right. So what PCR does is it's going to amplify very specific DNA segments. And it's a pretty amazing system. It's like a photocopy machine for genes. All right. And it allows us to start off with a very, very small amount of material and amplify it so that we have millions to billions of copies after just a few hours of doing this. All right, and so PCR is made up of 3 steps, and you can see them here. It's going to help you with your lab report. There's the first stage is what we call the denaturation phase. All right, and we're going to show this in a second. What happens in denaturation is that double-stranded piece of DNA. So let's think about DNA. It's, again, a double helix. Oops, let me go back. DNA is a double helix, right? And it's got 2 sides, essentially halves. And the halves are interconnected by hydrogen bonds. Now we know that hydrogen bonds are the weakest of all the chemical interactions. and they can be broken really nicely with heat. So heat, we're going to heat this up to near boiling. And what that's going to do is it's going to cause the hydrogen bonds that are normally keeping this double-stranded piece of DNA annealed. It's going to cause those two halves to come away from each other. So the longer you do it, the bigger, the more denaturation that occurs. And so we're typically going to do that, and it's going to break all the hydrogen bonds between the DNA strands. The next step, and I'm going to show you a video about this in just a second, it's called the annealing phase. So whenever you're doing these sorts of experiments, you kind of need to know what you're looking for, right? What is it I want to amplify? So for instance, in the COVID testing, they know what the ends essentially of the viral genes are. So essentially, what I mean by that, if you think about the alphabet being from A, B, C, D, all the way to Z, and if I wanted to amplify all the stuff that's in between A and Z, I can make a primer, and it's actually really easy to do for A, and then I'd make a primer for Z, and now I can literally amplify the alphabet, all the letters that are in between A and Z very easily. So let's think about this with a gene. If I know, again, what the beginning part of my gene is, I know what the end part of my gene is, I can now make primers that correspond to the flanking parts, the bookends of that gene. And now I can actually copy it. And this will make a little more sense. So usually those little primers are only about 5 to 7 nucleotides long. And again, they're called DNA primers. They're only about, again, 5 to 7 nucleotides long. And again, they flank the region of interest. So when we do that, what we're going to do again, we're going to change the temperature from 95, and the temperature drops down very, very quickly to about 55 to 65 degrees. There's a range. And what happens at that low range is that it gives enough time for hydrogen bonds to reform between these short little primers and the DNA of interest. All right, so that's what's going to happen very quickly in that middle region, that annealing phase. Now, very quickly, you're going to crank up the temperature to about 72 degrees Celsius. And what happens there is that in the PCR mix, we have an enzyme we use called Taq DNA polymerase. Now, DNA polymerase, as the name implies, is an enzyme that makes DNA. And this is a very special subtype of DNA polymerase that it comes from an organism called Taq. And Taq is short for a bacterium that was found in the geysers at Yellowstone National Park, actually. I don't know if you guys have ever been to Yellowstone. They have these really, really hot geysers. It's like boiling hot water that comes out at certain times, like at Old Faithful. And normally, all bacteria is not functional in boiling hot water, right? It actually denatures the proteins typically in those boiling hot temperatures. But if you go to Yellowstone National Park, there is literally a bacterium, a very unique bacterium called Thermophilus aquaticus. And scientists found that this bacterium lives in that environment. And again, it's a bacterium, so if you ever talk about it, you should either italicize it or underline it. And that this organism loves the hot water, right? Most things are denatured or destroyed in boiling hot magma water, but this organism has enabled an ability to survive in that environment. Now, why is this important? This is important because we need an enzyme that's going to work in hot temperatures, right? Even we're doing several rounds of temperature oscillations, right? The temperature goes from 95 to 55 to 72. And what we're going to see happens again is that it's going to repeat this. So this is going to repeat over and over again as we do PCR. And so we need a DNA polymerase that can withstand that boiling hot temperature that it's going to get to 95. It's very, very hot. All right, so let's look at a video of this. All right, so here is a video of this process, and it's a pretty good one. All right, let's just sit back and watch it just a couple minutes.
Polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands.
And again, that was accomplished by the hydrogen bonds that are normally holding the DNA into a double-stranded segment to create, the hydrogen bonds are all broken, and we get single strands.
When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
Isn't that cool? All right. Ooh, let me get out of this before it plays. All right. So if we go back to our PowerPoint presentation, let me just show you something. Hopefully I'm sharing it. All right. So what we see there is if we had just one copy of a gene that is amplified by PCR, notice what at the end of one round of PCR, you would have 2 copies. It says 1 copy, but I meant to write 2 copies there. Oops, there you go. And then this process, like we just said, is going to repeat. And so it's going to be an exponential growth of the product that's being produced each round. So after another round, we'd have 4 copies. Another round, we'd have, I guess, 8 copies. Another round, we'd have, so we'd just keep amplifying and getting up bigger and bigger and bigger. And so at the end, we would actually have billions of copies, not just millions of copies, billions of copies. So you have a huge amount of DNA now to work with. Now, this is why you can find a sample of one little piece of hair at a crime scene, right, a small sample of semen from a crime scene, and they're able to then amplify the genes of that individual such that we can now manipulate it and see what is your DNA fingerprint, right? Your DNA, like when you make a fingerprint, you have to have like enough of your finger there, right? You have to have enough ink on your finger to really make a fingerprint. And so when we're doing DNA fingerprinting, we need enough of your genomic material so that we can actually kind of see it, so that we can visualize it and determine who the individual is. And so typically what happens is that you would amplify certain genomic sequences that are kind of related to that fingerprint, right? There's certain regions in your genome which tend to be like kind of unique. And so that's what they're amplifying. So when we do these sorts of DNA fingerprints, all right? So again, we don't expect you to be a master at all this, but we're talking about the basic kind of concept of PCR, and you're amplifying specific DNA sequences, and then we're going to use them so that we can really look at them and determine who is who specifically. All right, so let's determine how we determine who is who. How you determine what genes are similar and what genes are different from 2 individuals is that we'll take some of that DNA sample that we've prepared and now treat it with what we call restriction enzymes. Now, restriction enzymes are unique enzymes that cut DNA at specific sites. And so I'm going to write that on there. Let me see if I can annotate this. Yeah, restriction enzymes are usually derived from bacteria. It's less important. And they were first identified in 1973, and this is what made genetic engineering possible. The reason why is they cut DNA at specific sites. All right, there's a lot of them. There's literally books of them. So the very first one that was identified was EcoR1, and the ECO stands for E. coli. That's the name of course, the bacteria that it's derived from. And the R stands for it being a restriction enzyme that cuts DNA, and it's number 1 because it was the very first one identified. It's Pretty cool. So what happens with EcoR1 is that whenever there is a GAATTC sequence, it has to be all 6 of those nucleotides in a row, is that the DNA gets cut, the double-stranded gets cut in this sort of staggered way. So it creates almost like a puzzle piece that comes away from it. So the DNA is now going to get cut, and now we have these sort of overlapping hangers, these little overlapping zones that can pull apart. So we get these, what we call, they're called sticky ends if you continue to go into genetic engineering. All right, so EcoR1 is the very first one. And so what we would do when we're doing these DNA fingerprints is that we would use a series of these enzymes. So we have all this DNA material, and now what we do is we cut it. And we cut it in now your DNA is going to cut a little differently from my DNA. And the reason for that is I've got slightly different sequences based upon my genetic ancestry, right? Every one of us is unique in our DNA to a certain extent. We have a lot of stuff that's in common, but there's a lot of stuff that's unique from person to person, right? You may have your DNA cutting here and here. And so what ends up forming, again, is a different way that the cuts occur in 2 different individuals. Again, no 2 people will have exactly the same cutting pattern, except for identical twins. It's very, 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 very similar with identical twins, but there can still be tiny little differences or polymorphisms, what we call them, that occur in even identical twins, just because, again, stuff happens and changes occur. But again, no typical 2 individuals will have exactly the same cutting pattern. That same logic, children inherit a lot of their cutting patterns from their mother and their father. So usually about 50% of the cutting pattern comes from mom, and 50% of the cutting pattern comes from dad. Anyway, it's kind of interesting. And what you end up creating, again, is a different cleavage pattern. And why that's important is we can use these different fragments now in gel electrophoresis to determine who's the same and who's different, right? Yeah, so hopefully that's starting to make sense. All right, so let's go on to the next slide. So actually, let's see. I don't know if we want to do the next slide or not. Let me get rid of this. All right. And again, each of us is different because each of us have tiny little differences in our DNA sequences that are caused by mutations, right? All of us have mutations. Some of us, the mutations are not, not a big deal, right? For instance, what's a good example of freckles, right? Freckles is a mutation that's not a real big deal. It causes a slightly different phenotype in the skin, but again, not a big deal. And then some mutations, of course, do have an impact. They can make the protein or the genes that the proteins that they're associated with not function properly. But again, all of us have various different point mutations, and that's why that cutting pattern becomes so important. All right, so what we're going to see is that now we're going to so let's think about what we've done. I'm just going to go ahead and get a new screen here, and hopefully I'm sharing that. Awesome. And so let's imagine I have my DNA. Well, this is Dr. Rossler's DNA. All right, and we've amplified my genes, right? So a couple targeted sites, and my DNA is going to cut, say, here and say here. All right, so what that means is that I would have this fragment, and then this fragment. I would essentially have 3 fragments that would form by that cutting pattern. So if I have a restriction enzyme cutting here and a restriction enzyme cutting here, and this is my PCR product, I would essentially get one tiny fragment. Let's imagine this one has 100 base pairs. Let's imagine this one has 200 base pairs in it. And let's imagine that this one has, let's say 500 base pairs. All right, so for a total of what, 800 base pairs. All right, let's think about Dr. Bud, right? Dr. Bud comes from a different genetic ancestry than I do, right? Some of you have him as a professor. He's a good friend of mine. All right, if we think about Dr. Bud's pattern, based upon his difference in genetic background, restriction enzymes, well, let's just say one actually, might cut it just once. So I have 2 cutting sites by those restriction enzymes because he has slightly different DNA based upon his ancestry. He might just have 1. So now he might have a fragment that forms that's like, let's see, what do we think? Should we make this about half and half? He might just have a 400 and a 400. All right. All right, so why is this important? Now we want to ultimately run it on a gel. And that's what your second part of your experiment is going to be about. All right, let's think about what these gels, so these gels that we use are called agarose gels. All right, and so agarose is essentially, it's almost like a Jello, right? I'm sure you guys have all eaten Jello at some point in your life, right? And that Jello is kind of loosey-goosey. It's slippery. It starts off, you know, kind of, you get it really hot and then you pour it into essentially a mold, right? If you've ever made a Jello mold or seen anyone make a Jello mold, all right? And we can actually make gels, agarose gels that once they cool down have a specific shape to them. And the shape that we often use looks like this if I'm looking at it from the side, right? What you're going to notice is that there is a well, a hole, and that well doesn't go all the way to the bottom, but it goes kind of, you know, most of the way through. And what we're going to do with this agarose gel once it cools down is we're going to put it into a chamber that's like this. It's like a plastic container. And again, I wish we could do this, and we may get a chance to do a very similar experiment in week 9 and 10. I apologize that, again, this happened to you guys, all right? We're gonna put that sort of Jello mold with that well into a chamber, a plastic chamber, and we're gonna put this, what we call a buffer, electrophoresis buffer, over it. And it's got some salt and some good stuff. All right, so I've got my electrophoresis buffer, and we're gonna pour it so it's all submerged, all right? Isn't that great? All right, and then what we're going to do is we're going to take this DNA that we've cut up and has all these various different fragments associated with it, and we're now going to put that DNA, we usually mix it with like a loading buffer that makes it kind of heavy. It's almost like a syrup. It's not syrup, it's a glycerin is actually what we use. So I'm going to take my DNA and mix it with essentially glycerin. Now glycerin's kind of like a heavy, almost like a syrup. And what I'm going to do, I'm going to say that I'm going to take my DNA samples and I'm going to insert it with a needle into this well. So you literally like, and usually the glycerin has like a color to it, like food coloring added into it, so you can literally see it going in. And so my DNA and that glycerin are going to sort of sequester into that well. All right, so again, this has all my DNA in here. And what we're going to do next is pretty fun. We're going to hook up this electrophoresis chamber to an electrode system. And you guys know if you've ever jumped your car battery, right? Think about the battery that you're using. There's a negative end, and actually let me change the color. Let's see, make this black. There's a negative end of your car battery, and there's a positive end. Now, it's really important that we have the negative end of the terminal. So here's my terminal, and I'm going to hook it up to an electrode. There's like a, what do you call it, a circuit that kind of connects to this, a plug that connects to this. And so the negative charge is going to go into here. And you know negative charges are going to be attracted to the positive that's going to be hooked up to the other side. All right. All right, so this is hooked up to an essentially almost like a car battery. All right, I know I keep saying that. Now, why is this important? We always want to have the negative end closest to the DNA to start off with. And the reason for that is the DNA, remember, is inherently negatively charged. So the DNA has a negative charge associated with it. Now, you guys know that charges that are the same don't like each other, right? So what's going to happen is that this negative charge in my DNA is going to try to get away from the negative, and he's attracted to the positive cathode over here. So what's going to happen in gel electrophoresis is that we're going to start to see these bands. The DNA is going to start migrating through the agarose, right, in a very specific way. Now these little bands are going to migrate, so hopefully that's making sense. If I could look at my gel kind of the other direction from the top, again, let's imagine I'm looking at those wells, this hole from here. Again, what's going to happen is that the DNA is going to start running down the length of the gel towards the positive end. Now, interestingly, these are going to run according to their size. So what I see here, let's imagine this is Dr. Rossler here. As the DNA is migrating, I've got this one fragment that's only 100 base pairs. That's really, really small. Conversely, I have this fragment up here that's 500 base pairs, and this fragment here that's 200 base pairs. Ask yourself, which of these fragments is going to travel the fastest through this agarose gel? What we're thinking here is that this fragment, hopefully you can start to see it, that 100 base pair fragment, he's going to be a really fast little fragment, right? It's like the person in traffic on the highway, right? You guys know that if you're stuck in traffic, the person on the moped or the motorcycle is going to be able to dodge and weave throughout the traffic. That's the same thing that's happening here with this 100 base pair fragment, is that because he's so small, he travels wicked fast through that agarose gel. There's little nooks and crevices in that agarose, and he's gonna travel the fastest. Let's think about which fragment in Dr. Rossler is gonna travel the slowest, right? This big fat 500 base pair fragment is gonna move wicked slow, right? Because it's like the semi truck that's stuck in traffic. So my 500 base pair fragment is gonna be over here. Now ask yourself, where do you think the 200 base pair fragment is gonna end up? Right? Somewhere in between, but probably right around here. All right, so when I do my DNA fingerprint on the specific region, I'm going to get a pattern after I've amplified my genes and after I've cut them with these restriction enzymes that looks a very specific way. I'm going to have 3 bands, and that is specific for me. Okay, let's look at Dr. Bud. Dr. Bud, let's put his DNA in this little well. All right, we know that he's going to have just 2 bands, a 400 base pair fragment and another 400 base pair fragment. So he's going to get one monster band. His is going to be really, really thick. And it's just going to be a little bit below where mine is for 500. And it's going to be nice and thick because he's got 2 copies of it, right? And he's going to get that one big band at 400. So this is how DNA fingerprinting works. And so we're able to identify a very specific gene or genes. We cut the DNA with these restriction enzymes, and what we get is a characteristic pattern after running them out on a gel that identifies one person from another person. And again, no 2 individuals are going to have exactly the same banding pattern, except for identical twins. It's going to be really, really similar. There might be just subtle, teeny, teeny, tiny little differences. All right, so now we're able to identify individuals or different genes from each other this way. All right, isn't that fun? All right, let's do this in a lab. So what you're going to do now is we're going to go back into Connect, and now we're going to do part 2. All right. And one little part I'm going to help you guys with as you're doing part 2, again, now the gene that we're going to be working on today is TNF alpha. TNF alpha is a really cool molecule, protein, and what it does is it is involved in your immune system. It helps to recruit immune cells to an infection site. So TNF alpha is a really important molecule in the immune system. Now what we're going to see is that there's 2 variants of TNF alpha. One is TNF alpha 1, and then the other, of course, is TNF alpha 2. If you have TNF alpha 1, you usually have a good immune system. But if you have this other version, TNF alpha 2, your immune system is not so hot, right? It's not able to recruit white blood cells as efficiently. So what we're going to see is we have a number of patients, and we're going to assess whether or not they have the good form of TNF alpha or the bad form of TNF alpha, and see whether or not their immune issues are related to that gene mutation. All right, so we're going to get a fingerprint for TNF-alpha-1, and then we're going to get a fingerprint for TNF-alpha-2 and see which one of these the patients have. Now, again, like we said, to do this, we took DNA samples from these patients, we amplified the specific areas with PCR, and then we cut them with restriction enzymes. So that's, again, called RFLP analysis. So cutting them with the enzymes after we kind of amplify them is called restriction fragment length polymorphism. Let me write that on here. So that was all called restriction fragment length polymorphism. Poly means many, morph means a shape. So we have many different shapes of our DNA once we cut it with these restriction enzymes. We're going to put them into a gel, and we said that the different size bands are going to migrate differentially. We typically use that loading die I was telling you about, and then this down here, I want to scroll. Oops. If we scroll down here, one of the things we often use too is we have to stain the DNA. And the thing that we use to stain the DNA is called ethidium bromide. And ethidium bromide helps to, it actually binds to the DNA fragments. And it's actually really carcinogenic. So if you ever work in a lab, be very careful when you're using the ethidium bromide. You have to use gloves and be very, very careful about it. And what it does though is it binds, it intercalates with the DNA, and it causes now the DNA to fluoresce under ultraviolet light. So we can actually see it. Now, we would, again, be doing this if we were doing this in the lab. We wouldn't be using ethidium bromide. We actually use a really cheapo, or not a cheapo version, a very expensive version that doesn't cause cancer because, you know, the university frowns on us, you know, causing cancer in our patients. Haha. All right, so let's go ahead and set this sucker up. All right, I'm going to help you with one little part. The very first part of this you may not know. Unless you work in a lab, you may not understand this. So what I'm going to do real quickly is I'm going to show you the different things that we would be using. All right, this right here is, well, let's do this thing first. This is remember I was telling you there's like a Jello mold that we use to make the gel, right? This is the Jello mold. All right, so it's a little plastic cassette. And we often use tape to bind the ends. So you literally will put a piece of tape on the front and on the back, and essentially you've made a Jello mold. Now we also mentioned that you're going to need to have wells or holes, you know, in the gel itself. So what we do is we place, this is called a gel comb, and we place it on top of the agarose gel case. And now we have essentially a nice little system that allows the gel to have a very specific shape. And then what we're going to do is we're going to pour really, really hot agarose. Agarose, again, is like a Jello. And we have to get it really, really hot in order for it to be a liquid. And then while it's hot, we're going to pour it into this case. And as it cools, it's going to solidify and then form that sort of Jello mold thing that we were talking about. All right. We're going to put that Jello mold, once it's chilled, into this electrophoresis chamber here. And like we said before, we're going to saturate it then with an electrophoresis buffer. And then we said we're going to take our DNA samples. To put the DNA samples, these very small amounts, we're going to use a pipette. This is called a micropipette. It does very small amounts. We're going to use a micropipette and a micropipette tip to take our DNA samples, which are all loaded over here, into those individual wells. So to make your life a little bit easier, and since again you may not know what all this stuff is, I went ahead and just figured out the answers, and I put them here. All right, so if you want, you can pause this and go ahead and fill all those pieces in so that you can move on with your lab. All right. And once you guys have all of that, I'll have you guys go through the whole lab protocol, and you're going to go ahead and, again, you're trying to figure out what DNA fingerprint each patient has for TNF-alpha. Do they have TNF-alpha-1 or TNF-alpha-2? All right. So go forth with all of that. All right, I don't know what your data is going to look like, but this is what my data look like. I don't want you to just take a screenshot of this, right? That's why I wrote on it. All right, so for your own lab report, you're going to get a picture at the end. All right, and hopefully it looks something like this. And go ahead and take a screenshot of that because you're going to use it in your lab report. All right. When you get to when you get that picture done, you're essentially kind of done with the lab. It's not a terribly difficult one. And then what you're going to do, of course, is you're going to use that information in our lab template. So I'm going to pull up the lab template on my side. All right, and that one figure that you generated, right, is going to ultimately go into the results section. There's not a lot of information in this lab that's going to go in the results, but that one picture of the gel that you just took, hopefully, you can cut and paste it and crop it and put it into that results period. And you need to have a figure legend underneath it that explains what patient 1, 2, and 3 ultimately are. So if you figured out that patient 1 has TNF alpha this, and the other patient 2 has this TNF alpha form, and patient 3, go ahead and tell us that in the results underneath, in the results section underneath in the figure legend. Okay, it should be very straightforward. You could also, if you wanted to, I think you had to do a table. You might have had to do a table, I think, for your data. You could cut and paste that table that you made in the lab into this results section as well, and that would answer all those questions. All right, but I ultimately just want to see the figure, the picture of the gel, and I want you to explain to us in somewhere what patient 1, 2, and 3 are. Now, the other stuff in this material, it's actually a very easy lab report. Again, what we were doing, the purpose of this lab was to determine the genotypes of TNF alpha in these 3 individuals using certain genetic or DNA tools, biotechnology tools. The biotechnology tools that we discussed, right? First thing we had to do was we had to extract the DNA. I want you to explain this in your own words. All right. And explain again specifically what the soap does, what the positively charged ions do, and why do we use cold ethanol ultimately to get the DNA. Again, you can cite me in your resources. Cite me as a source of the lab materials from BIO1016. But make sure you're citing all your sources as you move through. The next thing you're going to do is, again, just reiterate what PCR is. You don't need to spend a nauseating amount of time, but again, reiterate. You can use, again, the PowerPoint materials that were provided to you in this lecture and cite me or the sources. You could also go into Credo if you wanted to, right? You all are getting hopefully pretty good at going into Credo through in your library database system. And look at PCR. And then you're finally going to talk about gel electrophoresis, the idea that you have this agarose gel and the DNA is going to migrate through this agarose. And again, some fragments are going to move faster versus slower, and they're all according to their size, right? The bigger, the fatter the molecule, they travel slower. The skinnier the molecule, it travels faster. And again, cite your sources, it could be me. And then finally, the DNA fingerprint. How does restriction fragment length polymorphisms and doing these genetic fingerprintings work? It works because we take restriction enzymes and it cuts the DNA in specific ways, right? And we get a kind of unique pattern in the gel electrophoresis. So go ahead and explain all that. And here finally at the end, what would the restriction fragment patterns, why would the restriction fragment length patterns between the 2NF forms differ? They ultimately differ because, again, mutations occurred, right? There are slightly different mutations that occur in all individuals and slightly different polymorphisms or differences in those TNF genes that we were looking for by using this RFLP analysis. All right, hopefully that makes sense to everybody. And again, if you have any questions, just reach out to your faculty and yeah, and hopefully you guys can get that all turned in as soon as possible. This is a pretty easy lab and hopefully you felt okay. I know it's a lot of material in terms of background material, but the actual lab itself, hopefully we're gonna get a chance to do this a little bit differently in week 9 and 10. We typically do a PCR in week 7, and then we also do a PCR in week 9 and 10 that we'll get a chance to do if we go back to face-to-face, God willing. All right, take care, wear your mask.